The understanding of DNA has gone through large evolution, from its initial discovery as a molecular structure to its advancements in genetic research. Throughout the years, DNA has had large effects on our society and allowed for crucial medical advancements to be made. The history of DNA spans back to around 5000 BC, when humans began selectively breeding crops and livestock to improve their quality. This can be seen with the picture on the slideshow, as it showed how two crops are bred together to create the ideal one. Although scientific evidence was lacking, historical records indicate humans have always been aware and interested in heredity, and known that traits were passed down from a parent onto its offspring. Around 500 BC, people began to link this discovery onto humans and notice family resemblances in physical features, voice, mannerisms, and behavior. In later years, Aristotle, an ancient philosopher, began to make hypotheses about heredity. He believed that blood was crucial for constructing all parts of an adult body. While his idea was proved wrong in later years, Aristotle was one of the many important people in the history of DNA. Slide. While the initial ideas surrounding DNA were filled with strong discoveries, true advancements were not made until 1831, when Charles Darwin intended a research trip that later shaped the world of genetics that we know today. Darwin observed the natural world during the journey and formed the idea that life changes over time, with evidence found in fossil and living organisms. He discovered the changes he found in the species were the result of a process called natural selection, which involves DNA to change and evolve through generations. This can be seen with the picture on the slideshow, as it shows how one species of bird was able to change into many different variations as it separated and went through different situations. In 1863, Gregor Mendel used Darwin's findings to explain why certain traits are more common than others. He performed experiments on pea plants and discovered that when a yellow and green pea plant were crossbred, their offspring always had yellow peas, but in the following generation, green peas reappeared in a ratio of 3-to-1. To explain this, Mendel introduced the terms recessive and dominant to describe traits. Following this discovery, in 1869, one of the most important pieces of history in the world of DNA happened. Frederick Miser discovered the molecule we now refer to as DNA. He isolated a substance called nuclein from the nuclei of a human pus cell and identified it as a new type of molecule with equal significance to proteins. Miser's identification of DNA paved the way for many scientific advancements. One of these included Archibald Edward Garrod's discovery in 1902, where he was able to make a connection between Mendel's theories and a human disease. He studied a certain disorder and gathered family histories from his patients. He determined that it was a recessive disorder, which marked the first time a genetic disorder was linked to passed down traits. Slide. As the history of DNA unraveled, it hit a dark chapter for society in the early 1900s. The eugenics movement was made to improve the genetic quality of the human population through selective breeding and sterilization. At that time, it was believed that certain traits and characteristics could be promoted, while others could be eliminated, leading to a more desirable and superior society. However, the implementation of eugenics led to significant ethical concerns and human rights violations, as it often targeted marginalized groups, such as people with disabilities or different racial backgrounds. Please watch this video from 0 to 52 seconds. Slide. The history of DNA continued as different scientists made small but significant discoveries about DNA structure. Alexander found that the DNA backbone consisted of repeating phosphate and deoxyribose sugar groups. Irwin discovered that the ratios of DNA bases were constant across species. Maurice and Rosalind X-ray images revealed the DNA spiral shape. In 1953, James and Francis built upon all these discoveries and proposed DNA's ability to replicate and carry genetic information. After this insane discovery, in 1960, Peter and David found the first chromosomal abnormality associated with cancer. They realized a small chromosome was present in individuals suffering from a type of leukemia. In more recent years, many technologies have been produced using the discoveries made throughout history. A high-speed DNA sequencing technique was used for sequencing the genes and genomes of multiple organisms. The Human Genome Project was a research initiative that aimed to decipher the human genetic code, develop tools for identifying disease-related genes, and also address the ethical and social implications of genetic technologies. The CRISPR genome editing tool was a cut-and-paste system that presented many applications in research and therapeutic endeavors. All discoveries and technologies made in the history of DNA have opened up new possibilities for scientific research, medicine, and personalized healthcare. As these technologies advance, they held the promise of further creations that have and will continue to shape our society. Slide. Current DNA technologies are heavily based on past discoveries. DNA sequencing involves determining the order of the 4 chemical bases in a DNA molecule. This sequence reveals the genetic information contained within the DNA. It helps scientists identify regions that contain genes that regulate their activity. By analyzing the sequence data, gene changes that can lead to diseases can be detected. DNA cloning is the process of creating identical copies of a specific DNA sequence. It has diverse applications, including gene therapy, where healthy versions of genes are provided to individuals with genetic disorders. Gel electrophoresis is a method used to separate large molecules such as DNA, RNA, and proteins. As seen in the picture, it shows a diagram of gel electrophoresis, which shows how DNA samples are loaded into the wells at one end of a gel, and an electric current is applied to pull them through the gel. These DNA fragments are negatively charged, so they move towards the positive end. Current DNA technologies have brought numerous benefits to society, giving us deeper insights into our genetic makeup, providing better healthcare, aiding in scientific advancements, and addressing various societal challenges. Slide. While many aspects of DNA could be insanely helpful to our society, there are many ethical problems that come with it. CRISPR offers potential for treating genetic disorders like cystic fibrosis and sickle cell disease, as well as complex conditions like cancer and heart disease. However, the use of gene editing in human embryos raises concerns, such as experimenting on minors, destroying human embryos, and artificially creating life. The concept of designer babies blurs the line between therapy and enhancement, with long-term effects on future generations. Along with these concerns, cloning animals is a major problem as well. Cloning attempts for cats and dogs have a low success rate of around 25% and pose challenges such as failed embryo implantation, miscarriages, and the birth of animals with defects. Moreover, the cloning process causes harm to the animals involved. Offspring born with defects suffer during creation and are ultimately euthanized during their deformities. In other cases involving disabilities, many questions become apparent. These problems arise because there are no definite solutions in these situations. Some questions include, should someone be considered to have a disorder if they have a genetic defect but show no negative traits? And who has the authority to decide this? Moreover, is it morally right to prevent disabilities? These traits contribute to a person's uniqueness, and attempting to cure them may erase individuality and enforce societal norms. Furthermore, seeking cures for those with disabilities can cause psychological harm. We shouldn't undermine their experiences by trying to fix what society views as wrong with them. Ultimately, these questions remain unanswered, as there is no real right answer to any of them. While many DNA technologies could change the medical world, the ethical issues should always be our main focus and concern. It is very important to address these ethical problems and ensure that DNA technologies are used responsibly, equitably, and in the best interests of the individuals and society as a whole. That way, the promising future of DNA can be fully explored in a safe and responsible manner. Slide. This future could involve revolutionary technologies and large potential for the medical world. Gene therapy shows promise in replacing defective genes with normal ones to restore proper function and treat various conditions like cancer and autoimmune disorders. Along with this, it may be possible to anticipate an individual's genetic makeup based on observational traits like eye color and height. Additionally, while we currently have the ability to grow and transform cells into functional tissues and organs like bladders and blood vessels, we could potentially regenerate organs such as the heart, liver, lungs, and possibly even the brain. These advancements offer hope for finding cures and improving human health. The future of DNA is full of promise. However, as discussed before, it's crucial to address ethical, legal, and social concerns to ensure the responsible and beneficial use of these technologies. Through continued research, innovation, and collaboration, we can shape the future of DNA applications, opening up new pathways for scientific exploration and enhancing the well-being of humanity. The history of DNA has brought us remarkable insights, and the future holds even greater possibilities. As DNA technologies continue to advance, they will have a profound impact on society, transforming healthcare, agriculture, and various industries. These advancements will change how we understand and interact with the world, offering solutions to complex challenges and improving the quality of life for individuals and communities worldwide.